So my name is Dan Pierre. I'm the head of the Laboratory of Nanomedicine at Tel Aviv University. I'm here at the EMRS meeting talking about harnessing nanomedicine for therapeutic intervention.
Our lab is studying how to manipulate cellular function in order to generate novel therapeutic approach to treat mainly leukocytes-implicated diseases. And this is a huge field that includes blood cancer, inflammation, and viral infections. And we are quite occupied in each of those buckets. So everything that you have here in this blue labeling is a program that we currently work. And I will try to share with you at least some of our data for the past few years, and maybe we'll have an opportunity to discuss later what does it mean. So our lab is divided into two major groups, basically people from material sciences, and this is how we interact with this conference site. So generating drug delivery system based of naturally occurring biopolymers, mainly lipids, sugars, and proteins. Protein engineers that makes targeted ligands or antibodies or fragments that specifically target specific cell types. And on the other side of the lab, we have the biologists, so immunologists and cell biologists that are working on target discovery and validation via RNA interference libraries and immunology concepts. And the idea is, of course, to combine those two arms into one personalized approach that we will be able to tailor-made the appropriate vehicle, the selective targeting moiety, and of course the therapeutic payload. So I'm gonna tell you a few stories. I'm gonna start back at 2005. We had lots of interest with this gene named cyclin D1, which is a pivotal cell cycle regulating molecule. Now it's been used as a known target for cancer therapy, but I will show you some data that this is also a very interesting anti-inflammation or anti-inflammatory target. In inflammatory bowel diseases, such as ulcerative colitis and Crohn's disease, this gene is aberrantly upregulated in epithelial cells and in immune cells, in leukocytes. Now, its role in epithelial cells have been predominantly correlated to colorectal cancer, because again, the proliferation, it's actually being constitutively active there. So these are cells that become constitutively proliferating. But in immune cells, in leukocytes, it was really unclear why this is constitutively expressed there. So we set up a strategy to target those specific gut mononuclear leukocytes that home to the gut from the circulation and trying to silence cyclin D1 using RNA interference approach in an experimental colitis model. So we were looking for receptor targets for siRNA delivery to leukocytes. And there are many, many options. And one of the options are leukocytes integrins. Integrins are largest family of cell adhesion molecule that mediate cell-to-cell and cell-to-matrix interaction. And this is how they interact inside the blood vessels with endothelial cells. In those basically roles, the role of integrins is predominantly known. And there are a few reasons why people should use leukocytes integrins as their potential receptor targets to enter into the cells, and a few reasons why you should probably be aware of using there. So basically the beta-7, there's a mistake, the beta-2 integrins are exclusively expressed in leukocytes, and you cannot really find them in any other cells. They're readily internalized. So basically when the ligand binds to this integrin, it will internalize everything very, very fast, within 45 seconds in human T cells, taking up, for example, a natural ligand or an antibody with potentially nanoparticle on this. And some of them undergo conformational changes upon activation, and if I'll have time, maybe I won't, I will probably give you some examples. So it gives you another layer of specificity. And a word of caution, when you do basically using leukocytes integrins and you come with an antibody or a natural ligand, you're now cross-linking receptors. And this could potentially be a dangerous event because it could enhance the outside-in signaling events. And specifically, in inflammatory situation, you don't want to create an outside-in signaling. You don't want the cell to become activated or proliferate. So the expression of beta-7 integrins is quite wide inside the gut, using in a normal situation, and it's upregulated when you have an inflammatory bowel disease situation. So we have looked and we have seen that those, for example, a lymphoid organ named purse patch has some very unique T cells that express the alpha-4 beta-7. Intraepithelial lymphocytes within the gut area express the alpha-E beta-7, and of course in the lamina propria and mesenchymal lymph node, and there is a lot of crosstalk between those. The hypothesis that we had was that if we will be able to create a very tiny and effective delivery system, target those beta-7 integrins in the circulation, basically we will hijack the idea. Kind of a Trojan horse that we all talk about, but the idea was if we can target them in the circulation, we can deliver siRNA against cyclin D1, then it will knock down the gene, and maybe they will not be proliferating. Proliferating had that mean, at that time, also connected to migration. So the ability to migrate into the tissue. And I want to remind you, and if you don't know, I will explain this, that inflammatory bowel disease is a very complex set of diseases. And basically, leukocytes home from the bone marrow into the circulation and into the gut, penetrate and infiltrate into the gut and destroy the tissue. So we wanted to be in a position to silence the gene that is responsible for proliferating of those cells. Maybe the migration will be minimal and they won't penetrate into the gut. This was the hypothesis that was proven completely incorrect. So what we have found, so first I want to say that we published this. We had this seminal paper that we looked at, we generated lipid-based particles that are neutral in charge. We coat them with hyaluronic acid, and I'm going to talk a little bit about this more, as a scaffold. We put an anti-beta-7 integrin monoclonal antibody on the surface, and we package siRNA with human recombinant protein. But what I wanted to say that we have seen and shown that those particles, if we label the antibody on the surface with an Alexa 488 and the siRNA with the dye, such as Cy3, they penetrate very nicely into beta-7 expressing cells. Isotype control do not enter, and of course siRNA into leukocytes do not enter. So we have also shown that if we use a beta-7 knockouts, this will not happen, and this is under flow condition. This means that the specificity is very high, and the process is very, very short for internalization. We are talking about short minutes. Okay, we also shown that not only cyclin D1 is going down in an in vivo experiment, but something else is happening. For some unknown reason back then, we have observed that some of the pro-inflammatory cytokines in a very specific manner are also going down. This includes TNF-alpha and IL-2P40 units. And the anti-inflammatory remains the same. If this is correct, and we have verified with different sequences, etc., and it's all in the manuscript, if this is correct, it means that we now have a strategy to change the balance between pro and anti-inflammation. Okay, so basically, I'm not going to talk more about this, but we have shown that we can block this intestinal inflammation, okay, with this system. We have shown that this is what happened from the histology standpoint. This is colitis, basically, of the gut. So you can see infiltration of tiny, tiny dots here that destroy the tissue here. But then this is a normal gut section. So you see the cell, you see goblet cells, you see inside leukocytes, and the structure looks intact. And this is what's happened if we treat with the beta-7. We have all different controls here. But can we target those specifically? And can we use this for imaging purposes? So we hooked up with Alan Packard at Harvard Medical School, a children's hospital, and we used the same monoclonal antibody here with a copper 64, and we imaged this in mice that have colitis. And we have seen that about 7.5% of the ejected dose goes systemically to the gut tissue. And it's very specific because if we repeat the experiment and we use an isotype control, everything goes to the liver. So it's not going to the gut. So the idea to combine create a theranostic system of real-time imaging using PET-CT and therapeutics for leukocytes is something that is reasonably doable. Because many Crohn's patients undergo these days CT, at least 3 to 5 days, the sick, really sick ones, 6 to 5 times a year. So it's a lot of radiation, and if we can do molecular imaging going after the leukocytes, that could be an advantage, and you can do disease management. But RNAi for therapeutics is very, very challenging. And I actually got these slides, and I have to acknowledge my colleagues at Merck who gave me this slide. And you see all those big challenges that you have for a very negatively charged molecule, about 13.5 kilodalton, to enter really and work inside the cell. And in leukocytes, we know that this is even more sophisticated. I would like to focus a little bit about immune activation and toxicity. Back in 2009, a few clinical trials using siRNA and cationic liposomes were used in the clinic. And some of them was basically reported to induce flu-like syndrome in patients, at least in the higher doses. We thought that flu-like syndrome are basically cytokine storms. And we wanted to understand why does it happen. And to understand this and to understand the biology, we knew already that exogenous double-strand RNA from 30 base pairs and up actually activates antiviral defense in mammalian cells through interferon and NF-kappa B dependent pathway. But this was known. Also, the work published in Nature in 2008 by Joe Ambarti teaches us that siRNAs, small one, around 19 to 21 nucleotides, actually can do the same, or can antagonize, toll-like receptor 3, potentially also 7 and 8, in a sequence-dependent and independent manner. We also knew that the clinical trial also already mitigate this risk because they use modified siRNA, special chemical modification on the siRNAs, such as 2-methylated lock nucleic acid, etc. So we couldn't really agree that this was the real reason behind this. So what is going on? Do we see a nanotoxicity here? Or immunonanotoxicity? And in a set of paper, we try to analyze this. So is it the carrier charge, size, shape, composition, curvature? Is it the combination with the siRNA? What's going on? So very quickly, I have to show you immunology in a sense, I would say. Let's say we have a macrophage, we have toll-like receptors, it binds, this is toll receptor 4, it binds to LPS, lipopolysaccharides, basically contamination or something, and it's coming to secrete TNF-alpha and other pro-inflammatory cytokines, and this causes recruitment and activation of T cell and endothelial cells and other macrophages. But you have also a back feedback or a negative feedback where the macrophages start to express also and secrete IL-10, TGF-beta, etc., that blocks this signal and create an anti-inflammatory signal. Having said that, what we have done is to use neutrally charged, negatively charged, and positively charged cationic formulation with siRNAs, and you can see the zeta potential at those conditions, and they're all being around 100 nanometer in diameter. So basically, we thought that this is giving us the opportunity to test this. We use human fibroblasts because it was accessible to us, and we use an INSEL 2000 to do robotic screening, basically, and see what's going on. In a paper we published in Nanotechnology in 2011, we basically looked at different conditions in the cells. Basically, what we have found, it's very similar idea, that cationic formulation, and only the cationic formulation, kills the cells. To some extent, we don't understand what the mechanism behind this. And what's happened when you don't understand? Killing has lots of potential. We don't know if it's coming from a mitochondria issue. We don't know if it's coming from a Golgi issue. We don't know if they're suffocating the cell. We don't know what's the mechanism. So we went into in vivo and basically injected into healthy, normal C57 black 6 mice, and we found this phenomenon. Significantly, injecting cationic formulation decreased after 4 IV injection the body weight. But also, if you can see, these are normal healthy cells, they're normal healthy mice, so increasing their normal body weight. But if you inject neutrally charged or negatively charged, they're also going down. So something is happening here. We also decided to do a panel of cytokines and look to see what's going on. And we found, very surprisingly, upregulation of the cationic formulation in all the pro-inflammatory cytokines that started 2 hours post-injection and actually went down 24 hours post-injection. And also, if you look at interferon-responsive gene, basically this is an interferon response. They're going up. Here we had fears that maybe we have a contamination, lipopolysaccharide contamination or other contamination in our particles. So we repeat the experiments, this time doing everything under clean room conditions and with lipids and all the material that are GMP material. And we repeat the experiment, we get basically the same trend. This was very surprising because for a person that is half an immunology like myself, this looks like an LPS activation. So we did a killer experiment. The hypothesis was the cationic formulation agonizing one of the toll-like receptor, predominantly toll-like receptor 4. And to prove this, we used wild-type mice and TLR4 knockouts. We injected systemically siRNAs with nanoparticles, the cationic formulation or the cationic itself, and we sorted CD11B cells, which basically marks you monocytes, macrophages, and neutrophils. And we looked at different cytokines. For example, an anti-inflammatory cytokines like IL-13 did not do anything, but all the pro-inflammatory cytokines went up. Now, if we use the TLR knockouts, you see that they are going down. These bars are actually internal control. These are siRNAs that were reported by Joan Barty to agonize toll-like receptor 3. So this is an elegant experiment, and the mirror was here. So we revealed the mechanism behind this, and this made lots of problems to the pharmaceutical industry. Because now they said, we have a problem. And the problem means that, we need to be more creative. We need to be very careful when we use materials that are cationic, especially in clinical trials. We have to carefully go back and observe what's going on. And about 17 different formulations were tested clinically after this paper was published to just show that they're more safe. And the toxicity came from the carrier, potentially the carrier and the siRNA, and not the siRNA itself. And nowadays, we're in a position to say that if you look carefully at the literature, we are getting very positive response with minimal toxicity, which is a very good thing. So very fast, I'm going to go over this, just saying that at least our system is not inducing as the cationic formulation or the positive control. And I don't know if I have time to move also to other things, but another issue is the use of PEG. Now, we all like PEG, and we all are very happy with this. It's a long circulation molecule for us. But over the past few years, we observe lots of papers that basically saying that PEG can activate the complement system. Now, the issue here is that PEG is actually, if you put PEG that is 2% and above, it's a superstar in terms of the potential to activate human complement. And I'm not going to go into the details, but at the end of the day, we're talking about a potential anaphylactic shock. So we also tested this in our particles, and we show that at least the markers for complement activation, we done this with Moin Moghimi at University of Denmark, Copenhagen, from Denmark, basically are not activating the complement system. Now, how the immune system really sense or look at nanovectors? This is a problem that many people are trying to understand. This is the dogma, and I want to thank Janusz Zabini and Chezi Berenholtz for sharing with me this slide. And you can see that these should be in the lower level. It means that the immune system should not recognize them. But materials that are bigger are actually at the scope of recognition, or the triangle of recognition. Now, if you look at this, this is a cryo-TEM image, which is very interesting. This is doxil, pegylated liposomal doxorubicin, about 90 to 100 nanometer in diameter. This is HIV-1. It looks very similar. Okay? For some reason, these guys can activate the complement system. 5 to 7% of the patient do experience this essentially because of PEG. These guys has a lot of proteins on the surface and do not activate the immune system, at least by the time that those HIV particles are within CD4 cells, T helper cells. So what's going on here? This is a very crucial question for us. We're trying to understand really self, non-self versus danger theories, Poly-Metzger theories, basically versus the people that talk about self, non-self, and their own theories. And these are open questions. So I'm going to share with you some data and give you the opportunity then to think about those, because there is no proven experiment a killer experiment that proves either this or that, or the combination of the theories, which is now something that people do think. So together with Vince Rotello at UMass, we made tiny particles, about 2 nanometer in diameter, gold particle, with different Rs. And the idea was, let's see how the immune system is going to act if we have more and more hydrophobic moieties. So after injecting into mice, we have seen a correlation, for example, in healthy mice, that TNF-alpha is correlated with hydrophobicity in vivo. This made us think, what's going on? We all know that hydrophobicity could induce an immune response. But the question is, do we understand the mechanism? So we are working on this still. I'm going to leave you with this unknown question, and I'm going to shift gear to one project. Totally different from leukocytes, which is ovarian cancer. And we have a personal interest in ovarian cancer, and we are working on this for the past few years. And for us, it's not only a model for cancer, it's a model for a very aggressive and drug-resistant one. So, okay, the predominant mechanism for resistance are the efflux pump and predominantly P-glycoprotein. That is a very important mediator of the MDR. It's an ATP binding cassette transporter that efflux basically anything, usually small molecules, up to 1,000 Dalton in the molecular from different size, different chemistries, etc. We wanted to understand basically if we can bypass the PGP that is happening in those cancers. So we work with 2 cell lines. One is OVCAR-8 and one is NCI-ADR, which is actually, this is the parental version of this. It's also called NAR, which is highly resistant to doxorubicin. This is the expression by flow cytometry of the PGP, and it's actually effluxed up. And we used hyaluronic acid-coated lipid-based nanoclusters as our delivery system. We have worked with this delivery system for the past 6 years. It utilizes interaction with CD44 variants and the natural ligand hyaluronic acid, and we have shown in a set of paper what's the advantage of this. These are not liposomes and not micelles. Basically, we use phospholipids that do not form a lipid-based or bilayer structure, but clusters. And we can interrupt virtually anything from small molecule to nucleic acids. And I'm going to show you some data. We are utilizing the interaction between CD44 variants that are highly expressed in many tumors compared to the standard form that is expressed basically everywhere. And we have shown that hyaluronic acid binding, or those gagomers, as we term them, binds to those different variants and will not recognize or minimally recognize the standard form. And we have published a few papers showing from a paclitaxel, mitomycin, doxorubicin, nucleic acid, the variability that you can entrap in those systems. So going back, or before I go back to the ovarian cancer, I just want to show you something that we published a few years ago, which is very intriguing. This is from the operating room of patients that have thyroid cancers, and this is one example. In the paper we have a few others. And now if you take the same patient that has thyroid cancer on one side and the other side of the thyroid is cancer-free, and you make single-cell suspension of the thyroid sample, you can now stain them. If you stain with the pan-CD44 monoclonal antibody that recognizes all conformation, it could not recognize the cancers from the normal tissue. But if you take the natural ligand, and the high molecular weight is very, very important, it can distinguish, at least by flow cytometry, the cancer tissue from the normal. Now you think this is a big issue, but actually many adhesion molecules can do this. I've talked about integrins before, and one of them, LFA1, has a very known ligand, ICAM1, intracellular adhesion molecule 1. The interaction between ICAM to LFA1 is very similar to the interaction of CD44 variants to the natural ligand hyaluronan. It means that they can recognize only the activated form. They undergo conformational changes. Potentially, this is what's happened with the variants as well. We don't really know. But this phenomenon is something that happens. We also use some surface plasmon resonance analysis, putting the receptor on a chip and flowing those different molecular weights. And I'm going to tell you that size matter because, and I apologize for this slide, basically we have found that when you go up in the molecular weight from about 700 kilodalton and above, it's going to bind to the receptor without the ability to have a K off. It binds and opens new binding sites. Bind and have another binding site. And this is an issue. I mean, you can't really get it out of the receptor. Okay, we also gave some insight, and I'm going back now to the ovarian cancer, that if we now encapsulate doxorubicin in those delivery systems that we term gagomers, and if we knock down with siRNAs PGP, okay, we get much more drug inside these guys, which actually means that they're circumventing the PGP mechanism. Okay, just to show you some picture, these are the cells, NAR cells, without any treatment. You see those philopodia-like structure that they have. If you have those cluster of particles, again, I apologize for the image. You can see this at ACS Nano this year, about 2 months ago. You can see those clusters of particles that are around between 200 and 300 nanometers, but there is a lot of particles inside. So it's like I'll be careful, it's like a cluster bomb. It binds to the receptor, change the conformation. I'll show you data that it's penetrate and deliver the payloads. And it's a kinetic issue. So basically now you have more influx of the drug than an efflux. Very simple approach. And we, with the help of Professor Doron Shabbat, a chemistry guy in our university who labeled the Cy5, one of the lipids, DLPE, phosphatidylethanolamine, we can track those, and this is doxorubicin nuclear staining, and this is the lipid, one of the lipids of the particles. We can, in a Z-stack, we can see them inside the cells very elegantly. And we developed a model for this resistant tumor. We injected cells that have GFP expressing, stable expressing in our cells. You can track them already 14 days after you implant them. You can visualize them either in an IVIS or a Maestro, so it's easy. And if you take the tissue and you now do a single cell, you can actually fax them and you see CD44. This is the pan CD44, so all variants are here. And if you look carefully and you do histology, you can actually see the tumor, the subacute tumor in the skin. And if you do a blow up on the image, you can actually see new blood vessels are forming. So it's highly vascularized. And what we have done is a very simple PK study going after actually both the particles and the drug, which were actually correlated to some extent. But here, this is the drug as an example. And you can see here the doxorubicin plasma concentration that declined very, very fast. And again, these are tumor-bearing mice. And you can see the long circulation that these guys having. It's probably due to the shell, the hyaluronic acid, that is very hydrophilic coat. And then we also looked at quantitatively the percent of injected dose per gram tissue, and we found that we get in the tumor about 25-fold higher than the normal doxorubicin in the tumor, and we get much less liver uptake. And this is very surprising. Because even if you don't really know biology, you know that particles will be trapped in the liver, at least round particles. And larger particles probably are less trapped in the liver. Now, surprisingly, the liver is full of hyaluronic acid receptors. So hyaluronan is expressed, but also the receptors in the liver are highly expressed as well. Why don't we see them in the liver after a first IV path injection? Now, apparently, there are two issues here. The size is more than 100 nanometer, so predominantly it should be taken up, maybe by Kupffer cells or other macrophages, stellar cells in the liver. But also, when you have a binding, something could happen. So apparently, these were not activated CD44, only CD44 standards. And therefore, and again, what I'm telling you is based on other paper that hopefully will be published very soon. Apparently, it's very important. They are not uptake by CD44 standard, which is really expressed everywhere. So, okay, we also saw that it gives a predominantly an advantage. Again, this is a very aggressive tumor, and we wanted to compare different treatment. And you can see here, this is tumor volume basically without measuring this. So it's all electronic, measured by not an electronic caliber, but measuring by the imaging. And you can see that you can inject much less and get a very nice response, and the rest are going up in terms of the tumors. And even if you do half of the dose, you get it. So I just want to summarize this and tell you that we believe we know a lot of things in biology. It's much more complicated than we really know. I think that we undermine many things, including the immune system. The system I've just showed you was actually in nude mice. And in nude mice, we know that it's lacking T-cell activities and NK cell activities, B-cell activities. It has some innate immune activities, macrophage and dendritic cells, but it's not the same. So actually, if you want to look at good interaction, you should probably use, and this is a big argument in the field, which type of mice, but syngeneic mice that have a complete immune system, if you really want to look at immune system, this is probably the way to go. The best is, of course, humans, but this is a different story. So if we now summarize this specific work, I talked about targeting to tumor cells using a natural ligand of CD44 variants. We've shown this, and also in vivo, and it has enhanced drug accumulation, and it's more effective. I haven't shown you some safety issues, but it's all in the papers. And it has a versatility in the therapeutic payload. At the end, I just want to thank the people who really do the work. I'm actually just presenting, but I don't really do anything. So I think that the postdocs and the research associate and the graduate student that are behind the work here, and the funding agencies that support our work. And I want to thank you all for listening.